Hi, my name is Gregory Nyce, and thank you for giving me the opportunity to talk to you today about a serious issue facing our society. And it begins with a single plastic bottle. Bottle packaging has evolved over the years to make it cheaper and more convenient. But that convenience has a cost: plastic waste. Plastic waste impacts our environment, our food supply, and our health. To give you a sense of the scale of the problem, by the time I finish my presentation today, 10 million plastic bottles will have been purchased throughout the world, and it is estimated that over 500 billion plastic bottles will be purchased in the year 2020. One option to address this problem is to recycle, where the value of the new bottle is equivalent to the value of the used bottle. We have another approach. With the help of our proprietary upcycling technology, we view plastic bottles as an important feedstock to prepare valuable antimicrobial therapeutics, resulting in a 10 times to 100 times step change in value from a used plastic bottle to our antimicrobial products. So how do we go from a plastic bottle to an antimicrobial therapeutic? Well, first, we break down the bottle into its primary building blocks using our proprietary process. And then we add other building blocks and link the components in the mixture together to make an antimicrobial macromolecule in a simple two-step process. Adding different building blocks to the process results in different products with unique properties that can be tailored to a specific market or use case. Because we can readily tune the properties of our antimicrobial products, we view this as a platform technology that has the potential to address needs across many different markets. Before I discuss markets applications further, I'd like to go into more detail about our antimicrobial products and how they work. First, they're broadly applicable and effective against a variety of pathogen types. And they're potent at very low concentrations. They're highly selective for killing pathogens versus good cells. And finally, our products are resilient to antimicrobial resistance. Antimicrobial resistance occurs when microbes evolve mechanisms that protect them from the effects of antimicrobials. So unlike conventional technologies that become less and less effective over repeated uses, our products have the same efficacy after many uses, and we have not seen any evidence of antimicrobial resistance development using our products. This is a key feature since it'll make it easier to control and treat diseases now and into the future, and less product will be required to effectively manage disease. So how does our technology work? Our antimicrobials are engineered with two key features. First, the material is engineered with polymer charges along the macromolecular chain. These charges are attracted to the negative charges on the pathogen membrane surface. Second, once the macromolecule is on the membrane surface, elements along the chain can intercalate and disrupt the membrane, causing critical cellular materials to leak out and ultimately cause cellular death. So the properties of our antimicrobial compounds and their tunability enables us to address needs across many different markets. For today, I'd like to focus on the agriculture market and discuss a case study using our antimicrobial therapeutics in collaboration with a multinational beverage company. We estimate the total agriculture antimicrobial market to be on the order of $30 billion, and the global antimicrobial market we estimate to be $100 billion. There are opportunities to apply our technology in different agricultural markets for many types of diseases. I'm only going to focus on one disease known as citrus greening that resulted in a collaboration with a multinational beverage company and highlights our technology and how it can solve real-world problems. So the primary motivation for the collaboration is that the company's multi-billion-dollar orange juice business was under threat from the citrus greening disease. And the current solution, which I'll refer to as Compound X, it works, but it has a lot of undesirable attributes, and the company was looking for alternative solutions. In addition, the company uses approximately 120 billion plastic bottles per year, and our technology that repurposes plastic bottles to make antimicrobial compounds and also requires low dosing amounts to treat plant disease aligns with both their internal sustainable packaging initiative as well as their sustainable agricultural initiative. So what is citrus greening? Citrus greening is caused by bacteria that attacks the citrus tree's vascular system. It damages the fruit as shown in this picture, and it ultimately kills the tree. In Florida, there's been significant decreases in citrus fruit production over the past decade due to this disease. We did a head-to-head comparison of the company's current solution, Compound X, compared to our antimicrobial therapeutic, and here are a few key takeaways from that study. First, our product vastly outperformed Compound X. It was 100 times to 1,000 times better at killing the citrus screening bacteria. Now I'd like to highlight a few environmental considerations. Our compounds will decompose in the environment much faster than plastic bottles, and it is estimated that a plastic bottle will take roughly 450 years to fully decompose, while our compounds are estimated to decompose in the environment in a matter of months or less. As I mentioned earlier, because our products are AMR resilient, less product will be introduced in the environment while still effectively managing plant disease. And finally, the results of a metagenomic study that we undertook suggest that no change in the soil's microbiome occurred after repeated application of our compounds. And so there's no indication that our compounds would be harmful to the environment. Our technology and approach result in several competitive advantages. First, our products have a broad field of use and are effective against different types of pathogens, resulting in simplifying disease management for the end customer. Because our products are resilient to antimicrobial resistance, fewer treatments and/or less product will be required to manage disease. Our technology is scalable and can be tailored to a specific use case with the potential of lower cost and better targeted performance. And finally, our targets are derived from waste plastic, which we feel will be looked upon favorably by customers and consumers and may even command a green premium price in the market. We have a robust patent portfolio and it continues to grow. And we have a great team with over 50 years of combined experience in chemical, polymer, and chemical process development. At our core, we are a chemical technology company, and so our focus will be on the research and development of chemical technologies and protecting those technologies with an appropriate IP strategy. Once a technology has reached sufficient maturity level, we plan to then contract with the CMO to produce our products. Our products will pass back through us to sell through our target markets through an appropriate distribution channel. Our ask is an initial $3 million investment, and we plan to use these funds to scale up our antimicrobial products to pilot scale for seeding the market, finalizing a conceptual process design to prepare for contracting with CMOs to make our product at scale, and continue to grow our product pipeline. Managing plastic waste is one of the grand challenges of this century. Our technology uses plastic waste to yield valuable and effective antimicrobial compounds. Our compounds will make disease easier to treat and manage across a variety of markets. Investing in this technology will result in significant value creation with a strong social impact. Thank you for your attention, and I look forward to answering any questions.